Hello everybody, I am Dr. Farhan Zamir, an adjunct professor and academic expert at Biotecnika, Bangalore. At Biotecnika office, we get a lot of queries from science enthusiasts and from technologists asking the same question. The question is, sir, I know a small component of biology, how do I upgrade it? And the next question from the technologist is, sir, I also understand some component of computers. How do I implement this technology of computers onto biology? So the answer for this is the same. The thing is, you need to learn bioinformatics. And now, when I talk about bioinformatics, people might think there's a lot of computers which are involved, and the computer technologist could think there is a lot of biology which has been involved. In this video, we will try to see how comfortably we can try to understand biology so that it makes a huge sense in terms of technology. So let's dive in. The topic for today's discussion is the basic bioinformatic concepts which a beginner should understand. The video has been made in an intention in such a way that a PU passed out or a person who is from a non-science background can also understand biology well. So this video is mainly dedicated for those students who wants to understand technology in terms of biology background or in terms of technological background. If they want to understand biology, this video is dedicated for you people. Let's try to dive in. Now, before we see what exactly is bioinformatics, let us try to understand what is bioinformatics. Now, to make sense about bioinformatics, we would go into this slide, which simply talks about the amalgamation of life in terms of technology. So for me, life is maybe human, could be bacteria, for maybe plant or a microbe or whatever. Now, if I am trying to dwell or if I am trying to understand biology in terms of technology, in terms of computers, wherein I am using computer as a tool to understand biology. So this is a subject, what we call it as bioinformatics in crude terminologies. So just to understand, it is an interface or it is a bridge which bridges both biology and technology, especially computer sciences. So here I am still trying to see the biological data, but however, this comes out of a phase which is the computer. So this is an interface between biology and computer sciences. So if I want to look or understand more about bioinformatics, so if I decipher the word bioinformatics as bio, info, ma, t and cs, which could be extrapolated into bio is biology, info refers to information technology, ma refers to mathematics, t refers to TICS, that is statistics, and then cs refers to the tool itself or the discipline itself, which is computers or computer sciences. So the important component is to understand life. So what is life? So life is fundamental biological processes which involves three major functions. The first function is growth, survival and reproduction. Anything which shows all these three phenomena are being considered as living. Now, who is actually supporting this? This has been predominantly supported by molecules which has a biological function, and hence we call it as biomolecules. These biomolecules are segregated into four major types. The four major type is carbohydrates, proteins, lipids and nucleic acids. Now, when I talk about carbohydrates, carbohydrates are the fundamental biopolymers. Proteins, lipids, nucleic acids, all these are fundamental biopolymers. And to understand this better, let me try to make you understand this analogy. And with this analogy, we will try to move forward. Now, what is a polymer? polymer is a huge chain. So when I have a huge chain, how is this huge chain built? This huge chain is been built with smaller units, and these smaller units are called as monomers. So I have two major components to be understood. One is a polymer and the other one is monomer. So if I try to use this logic or analogy with a construction of a house, how is a house constructed? A house has been constructed with small bricks with various colors or various dimensions. So these are the fundamental units. So these fundamental units are called as monomers. So these bricks, when they are put together, they make up a wall. These group of walls, when they come together, they make up a room. And this entire room or a combination of these many rooms will make up a house. So basically what we are trying to understand is I have a monomeric unit, that is my brick, and through the combination of these bricks, I am trying to build up a house. So here, applying the same logic, I am trying to make you understand what is metabolism. Now, metabolism is a sum total process of the construction of a house or a destruction of a house. So here, metabolism involves two major steps. First is anabolism, and the second step is called as catabolism. Now, what is anabolism? Anabolism is a constructive process wherein these monomers will come together to form a higher molecule which is called as polymer, and this process of formation of a polymer is called as polymerization. And this basically happens with a process in metabolism which is called as anabolism. So remember, anabolism is a constructive process. Now, coming on to the catabolism, catabolism is a process wherein a huge polymer is been broken down towards a smaller monomeric unit. So this process is called as catabolism. So example, if I take up a house and I already have a constructed house, and if I want to have a renovation of it, then I am breaking the house into the smaller bricks, and this process of destruction of a polymer is called as catabolism. Now, anabolism and catabolism together defines metabolism. Now, with this as our basic understanding, let us try to understand the first sub-biomolecules, which is called as carbohydrate. Now, carbohydrate being a bigger polymer, this bigger polymer can undergo destruction, that is, there is a catabolism, and lead into glucose. Or this glucose molecules can come together to form a bigger molecule which are called as polysaccharides or carbohydrates. This process is called as anabolism. Now, how is this possible? This is mainly possible because of the combination of one glucose molecule with another glucose molecule with a bond, and this bond is called as glycosidic bond. Now, glycosidic bond is a bond which bridges one glucose unit to another glucose unit, so that a chain of glucose unit will then develop a macromolecule, and this macromolecule is called as carbohydrate. Using the same logic to the other biomolecules, the second example would be protein. Now, protein is a huge macromolecule, and anabolism or catabolism can lead into monomerization or polymerization, wherein the degradative process of protein catabolism can lead into the formation of a monomer, and these monomers of proteins are called as amino acids. One amino acid binds to another amino acid with a bond, and this chemical linkage is called as peptide bond, or it is also called as amide bond. Now, we'll try to understand another important macromolecule which is called as lipid. Now, we understand the fatty acids are the monomers, and these monomers will undergo anabolic reaction to form a bigger polymer, and this bigger macromolecule is called as lipid. However, the same lipid, which is a polymer, can undergo degradation to form a smallest unit which is called as fatty acids by a process called as catabolism, and the linkage which facilitates the formation of fatty acids to a bigger lipid molecule is nothing but your ester bonds. So the fourth biomolecule is nucleic acids. So nucleic acid as a bigger polymer, when it has been broken down or when it undergoes catabolism, it forms the smallest unit which is called as nucleosides or nucleotides. However, one nucleoside is being bound to another nucleoside with a bond, and this bond is called as phosphodiester bond. Now, I seek your attention on this particular slide or this particular picture. Here, the nucleosides are at the center, this which is ATGC, these ATGCs in a particular combination, it forms a set of codons, and this set of codons will actually bind for amino acids. So what I'm trying to make you understand is from nucleic acids, I am able to get amino acids. And remember, these amino acids are in a position to produce certain carbohydrates, or during my diet itself, I am able to produce certain carbohydrates. So the important point what I want to stress upon is we cannot look as these biomolecules as separate entities. They are all working together. Until now, the entire slide was easy and understandable. But however, once I understand the background of biology, the biology is not that simple. Okay, so until now we looked upon there is a macromolecule, there is a monomer, there is a polymer. But however, when I say that they are involved, the word involved refers to a big circuit. The big circuit involves carbohydrate metabolism, nucleotide metabolism, amino acid metabolism, lipid metabolism, and carbohydrate metabolism or secondary metabolism. So what I'm trying to stress upon is we cannot look at the biochemistry of these biomolecules as a separate entities, but however, in a living system, there are 1,000 reactions which are already going on, and understanding this at a molecular or sub-molecular level becomes very, very challenging. And here comes the important role of this fantastic subject, which is called as bioinformatics. So in bioinformatics, what do we learn? So let us try to illustrate with an example. Suppose I have my kitchen, and in my kitchen I have salt. This salt is nothing but NaCl. So the NaCl is nothing but Na plus Cl, which is a compound. Now, the studying of NaCl, which is in a box or in a jar, which is made up of either glass, or it is made up of steel, or it is made up of porcelain, whatever, according to your convenience. Now, what I need to study is how exactly the salt is reacting with another salt grain, or how exactly the salt is reacting with the jar in which it is being stored. So here, when I talk about silica or glass is nothing but silica. Now, the interaction of NaCl with silica can also be studied. So basically what I am trying to study is I am studying the physico-chemical properties of NaCl, and that branch which deals about the physico-chemical property of molecules is called as chemistry. When I talk about chemistry, here chemistry, wherein onto chemistry we can apply computer. So when we apply computer technology onto chemistry to understand this particular interaction, wherein I already have a chemical molecule, and how exactly the chemical molecule is interacting with my biological system, say for in this case any protein. So this is a specialized branch which is called as chem informatics. However, when I look into the other branch wherein I am good at understanding chemistry, so until now I was trying to give you an example of the salt which is stored in a jar. But now I do not want to take up that example, wherein now is a different situation. I still have that salt, but I am making you eat around 5 grams of salt. So now the salt does not remain as a molecule. Now what has happened there? The salt has entered into my system. My system is a biological system. So the salt from a molecule has been transformed into another molecule, which is called as biomolecules. Now studying the salt chemistry in a biological system is called as biochemistry, wherein now I am not trying to understand Na plus Cl or NaCl or NaCl with another NaCl or NaCl with silica. What I am trying to understand is how exactly NaCl interacts with my living system. Say for example, it can raise my blood pressure, it can lead into vomiting, it can lead into nausea. All these conditions is happening into a biological system, and studying that component is called as biochemistry of my body. However, to understand this, there are 101 reactions which are already going into my system. So I have a lipid layer, I have a carbohydrate receptor on it, I have cholesterol on it. So how exactly the salt molecule making a difference in my body? Either it could be a direct interaction or it could be an indirect interaction. Studying this becomes very, very important, and here comes a new opportunity to learn biology using computers, and this branch is called as bioinformatics. Now, This picture you would have seen from your 6th standard or 7th standard, wherein this is where the interaction of your molecule is being defined. There are two major theories which actually defines the interaction of a molecule with a target. So here, whenever I take up a molecule, I consider that molecule as a ligand molecule. So the ligand molecule is a molecule which is I need to test upon, and then I have a target molecule. Now, what is a target molecule? The ligand molecule interacts with some major biological molecule in my system, and that is what I consider as a target or a receptor. So when I lead into this particular reaction, so what would happen? So the ligand molecule will come and bind onto the target molecule, wherein if there is symmetry, so there is a beautiful conformation which happens. So you can actually imagine a lock and a key. So wherein not any key can open up an individual lock. So there is an exact complementary between a key and a lock, and that is how the key enters, opens up a particular lock. The same is the logic in biology. So any particular ligand should have a complementary configuration or conformation which can actually lead into the binding of a ligand molecule to a receptor, forming ligand binding complex. However, this is not the ideal case every point of time. So to define in a different scenario wherein you have an active site and the substrate is not complementary to that of a active site. So what happens here is there is another theory which is called as induced fit theory, which actually comes into picture, wherein the ligand molecule or the substrate molecule can go under modification. Okay, and this substrate molecule, depending on the active site undergoes modification, and that is how the substrate is being induced, and it will get fitted into the substrate. So this is how there is an ligand receptor interaction in terms of induced fit theory. So in induced fit theory, the active site actually modifies itself according to the complementarity of the substrate. However, in lock and key theory, it is a rigid model wherein the ligand and receptor has to get complemented with each other. This is what happens in bioinformatics or chem informatics, wherein we have a target and we have a ligand molecule. So this target could be any of the receptor, hormone, DNA, RNA, whatever you want to mention. Okay, that could be your target, or in this case, we have taken up a receptor molecule and we have designed a ligand molecule which is a chemical entity. So according to lock and key theory or according to induced fit theory, there is an interaction of ligand and receptor, and this interaction has led into ligand receptor complex. Now, in bioinformatics, the beauty is, it is not just I am trying to say that a ligand binds to a receptor, but I can very specifically say that where exactly my ligand is binding to the receptor. And you can see on the background of a protein, I have a ligand molecule which is binding there. And it is not just binding, I can also very specifically say where exactly or which exactly amino acid it is interacting with, and then what is the energy changes which is actually taking place, what is the hydrogen bond or what is the van der Waals forces which are actually involved, what is the bond length, what is the bond energy between this interaction, and very importantly, what is the atomic contact energy level between the ligand and the receptor could be identified, could be defined, and this is the power of bioinformatics. So to understand this, Lauren et al. 2022 has published a very good paper in Scientific Reports, which is a Nature Communication publication, and here they have explained the total applications of bioinformatics in biology. So here they have taken up an entire pipeline, and this pipeline talks about the metabolic data and then a gene expression data. So you can take up a pipeline, and in this pipeline, how can you process a particular data? Now, very importantly, you can annotate using bioinformatics. You can actually look into the filters and selections using bioinformatics. And then very importantly, you can either, depending on the fold significance or the expression level or the p-value, you can integrate, or you can look into the correlation between a gene metabolite or a gene protein interaction, and nevertheless you can always produce correlation networks and with the biological information, and you can produce regression models leading into the heat map which could express the upregulation or downregulation of the gene. I request you people to go read this paper and have better insights. The same paper talks about how exactly the information what we have got from the pipeline could be used for other wet lab experiment, and in this they are trying to develop a metabolic interaction between acetylcholine esterase and the other biomarkers which are present using KEGG databases. Now, with string or with cytoscape data, they are trying to look into how exactly these particular biomolecules or the substrate and the receptors are interacting, and this is through the cytoscape mechanism or the cytoscape database. And very importantly, apart from just looking into their interaction, this paper also talks about what is the biological function. So trust me, my dear friends, just taking up a protein on a virtual platform, that is using in silico platform, you can also define the functionality of a given protein. Now, with this as a dry lab data, okay, one can be assured that my wet lab experiments would be 80% true. And then very confidently, I can go into my lab and start my wet lab experiment. When the same thing happened with Loren et al. Paper, so they were able to look upon a cholinergic neuron wherein GFP, that is green fluorescent protein, was attached to it, and then the data which was expressed from the bioinformatic data was being able to replicate in their wet lab data, and that is how they were finally able to quantify using graphical representations. Now, the entire story put into one slide. So what I need to do in bioinformatics or this particular pipeline, this is how when you go into a pharma, okay, or with a pharmacist, and how do you get a drug? This particular drug has to follow or pass the entire pipeline, and only then you see it in the shelf of a medical store. So here, the first thing is you need to first identify a disease. Once you identify the disease, you need to identify the target. So target identification is very, very important. Now, this target could be a protein, this target could be a carbohydrate, this target could be a nucleic acid or any lipid molecule. So once there is a proper target identification, then you will take up a set of ligand molecules and you will screen them against this particular target to produce something which is called as hit identification. Once you produce this hit identification, you will downstream it to one major ligand molecule, one major biomolecule, which is of your major interest. And this step is called as lead optimization. Once you know the lead, so you are super confident with your bioinformatic data, that is your dry lab data. And now what you do is you will go into the wet lab studies. Now, wet lab studies could be done using in vitro models, in vivo models, okay, in situ models or ex situ models. So that component is called as preclinical trials, wherein either I do in vitro models or I will follow in vivo models, and then I will try to see what is the effect of my ligand molecule in living animals or into the chemical reactions. Once this qualifies ADMET, again, ADMET is adsorption, distribution, metabolism, excretion and toxicity, all these 5 phases which have been cleared in preclinical trials, then your drug is ready for the clinical trial. So this clinical trial normally has been done on a population which is called as YOPI, that is YOPI. Y stands for young, O stands for old, P stands for pregnant ladies, and I stands for immunocompromised patients. So once we look into all this, and if the drug qualifies your clinical trials, then the drug has to go for various kinds of drug approval and accreditation. And once it has been accredited by international and national agencies, only then the drug is available for you in the market. And this is the pipeline in which a drug travels from 0 to 100, and many a times on a slide it looks beautiful, okay, and it seems as it is very easy, but this is not the case. It takes billions of dollars to execute this particular work, and very importantly, lot of time and labor are being invested. And trust me, my dear friends, around 90% to 95% of the drugs actually they fail into clinical trials, and many a times, these are called as the failure drugs. So this is how research actually is been empowered, and the 10% drugs which actually comes out, will also lead, will have lot of hurdles in drug approval component. And if the molecule is then lucky, then it goes into the market. So this is how the entire journey of a molecule from your lab to the bench, okay, or from the lab to your medical store normally happens. And this is how the drug discovery and development pipeline works. Now, you learning bioinformatics will help the future in designing better drugs and better therapeutics, which can lead into a new drug discovery. And the intention of Biotecnika is to make you understand, is to make you, think, create, innovate newer drugs for a better future. And this is how the entire intention of Biotecnika is to make you the better scientists so that you can think well. And with the practical exposure, rather than just being into one domain of biology or one domain of technology, we want you people to have a combination of both biology and technology so that we can think better, innovate better, and create newer things for a better future. So thank you very much for your time. And if you have any suggestions, please do not forget to comment into the comment window, and also do not forget to subscribe Biotecnika. Thank you very much. We work with one major mantra, and that mantra is your success is our priority. Thank you very much.